Hello, my name is Simon, a graduate student from McGill University, and today I will be talking about modeling gene expression dynamics from single-cell RNA-seq data. Currently, for single-cell RNA sequencing, many algorithms exist for inferring pseudotime trajectories from the data. And RNA velocity is one of the interesting ones because it has the ability to infer the rate of transcription from the data. And cell trajectories can then be inferred from a Markov diffusion process. Scribe is an algorithm that extends upon RNA velocity, where it can infer the causal gene regulatory networks from time series data. And a natural follow-up to causal network inference is to model the system dynamics. And two of the methods that I'm interested in are the neural ODE method, as well as the ISMB algorithm. The neural ODE method is a neural network-based approach that directly fits an ODE to the data using neural networks as a universal function approximator. On the other hand, we also have the ISMB algorithm, which stands for sparse identification of nonlinear dynamics, that also fits an ODE to the data. The main difference between the two is that the ISMB algorithm restricts the set of functions of the ODE to be ratios between polynomial functions, whereas the neural ODE method typically can take on any function. And one thing that the ISMB algorithm can do is that in their paper, they showed that it is able to learn the exact equations for the data generating process and make out-of-sample predictions. So building upon the currently available methods, the overall aim of this project is to construct a scalable model that learns the underlying dynamics of gene regulation from time series data, with a focus towards pseudotime trajectories constructed from single-cell RNA-seq experiments. By combining and extending upon different computation methods available, we aim to construct a machine learning model that is able to extend the system dynamics over time and predict perturbation response of the system for events such as gene deletions. And more precisely, I will be modeling the system as a set of differential equations where the state variable x here represents the expression level of a collection of genes to be modeled, and the function p of s represents some user-defined perturbation of interest, where in the case of gene deletions, we can think of the perturbation as being setting its expression value permanently to 0. So in this presentation, I'm going to first show that our approach is able to learn the gene dynamics of a neuron differentiation pathway from single-cell data. And then I'm going to briefly talk about a first set of results on perturbation response based on simulated data. So first, to see whether we can extend the system dynamics over time, we use the neural ODE method to try and fit a differential equation on single-cell data for the neuron differentiation pathway. So in this case, we used cell trajectories constructed from single-cell data to try and learn the dynamics of 995 most variant genes. In this case, during the training process, we hide a part of the data along the trajectory and see how well the model performs. And once the model is trained, we give the model the initial states of the system and ask it to predict the time evolution of the system. And here is the result for this experiment, where on this plot, to characterize the overall performance, we looked at the convergence of the model towards the final state, where in this case, the x-axis represents the pseudo-time, and the y-axis is the L2 distance towards the final state. And each point here represents the L2 distance between a cell along the trajectory towards the final state. And the red portion of the data is the validation data, while the green points are the training data. The blue line represents the model predictions, and the black line represents a smooth line fitted to the data on the graph. Then we also plotted some of the genes known to be involved in the differentiation pathway, where in this case the y-axis now represents the expression level of that gene. As you can see, overall the model has a decent performance in capturing the dynamics along the trajectory as well as extending the dynamics into unobserved regions of the data. Then we also hide different portions of the data and evaluated the model performance. And we see that removing different parts of the data has somewhat different impacts on the model. So moving on to perturbation response, in this case, we first ran a simulated study to see whether or not our construct has the capabilities of predicting the effects of perturbations, such as gene deletions. In this experiment, we used a 4-variable system where the differential equation of the system are shown above. We first trained the model using time series data generated from 3 different initial conditions. And then once the model is trained, we evaluated their performance by looking at their predictions on slightly different initial conditions, as well as how well they perform on predicting the effects of node deletions. In this experiment, we also tried two different versions of the neural ODE method, where in the first case, we just trained a normal neural ODE on the data, where in the other case, we assumed that we have a causal network of the system, and so we can impose the network restrictions on the model. So in the network restricted version, we are now restricting the inputs for the computation of each node to only its neighbors, and see whether or not it helps with the model performance. And in the future, network connection data will be either obtained through prior knowledge or causal network inference algorithms, such as Scribe. So after the model were trained, we first see how well they perform on a different set of initial conditions. Here I'm plotting the values of the 4 variables over time, where the blue line is the true solution, and the other two are the model predictions. In this case, we see that both models performed well. Then we simulated the perturbation response, where we delete nodes one at a time by setting their values permanently to 0. In this case, we see that once we start deleting nodes from the system, the periodic behavior disappears. And in most of the cases, the graph-restricted version of the neural ODE method is better at following the true solution compared to its non-graph restricted counterparts. However, in some scenarios, both methods still fail. And here is the result for the other two deletions. So for future works, what I will be doing is to extend upon the current construct, where I will be extracting the cell trajectories using RNA velocity, and extending upon neural ODE methods, as well as the SIMD algorithm, and combining them with causal network inference algorithms such as Scribe to create a scalable machine learning model that learns the system dynamics of gene regulation and predict perturbation response. And hopefully in the future, we can use this model to investigate optimal intervention strategies for controlling different biological processes and disease mechanics. And lastly, I want to thank our collaborators, Celine and Paul, for helping us with this project. Thank you.